Thank you for the kind introduction. My name is Dr. Josephine Aimiuwu, and the purpose of my presentation is to address the common issues identified in in vitro release and permeation studies submitted in ANDAs to support bioequivalence for topical products. Our standard disclaimer that this presentation reflects the views of the author and should not be construed to represent FDA's views or policies. The learning objectives for today's talk are to recognize the role of IVRT and IVPT to support BE and its contribution to the submission of ANDA for topical products, outline the key attributes of these two in vitro tests. I will identify some common challenges across these studies, describe points to consider and the expectations from the agency, and finally provide a summary of my talk. As you know, quite a few of the ANDAs, FDA's current PSGs for topical products now have the in vitro option in addition to in vivo clinical endpoint and or PK studies. The in vitro option generally includes the assessment of the formulation, physicochemical and structural characterization, as well as the in vitro release and or permeation studies, which are the focus of today's presentation. In general, as the complexity of the dosage form increases and the potential for failure modes for supporting bioequivalence intensifies, the scope of the in vitro studies is likely to increase. And such, the recommendations can either include IVRT or both IVRT and IVPT studies. This in vitro test serves as performance test for assessing sameness between a proposed test formulation and the corresponding reference product. And finally, we know that IVRT describes the rate of release of API, while IVPT is the rate and extent of API availability at or near site of action. This comparable rate and extent can help mitigate risk for potential failure modes for establishing BE that are not adequately captured by formulation comparison and physicochemical characterization. This slide provides a snapshot of the increasing trend of topical product submissions containing IVRT and IVPT studies and the type of dosage forms in those submissions. As seen from the graph to my left, since 2010, there has been a steep increase in the number of ANDAs with either IVRT and or IVPT studies. This may be due to a better understanding of this in vitro test and the agency's publications of PSGs containing in vitro options for topical products. The chart to my right illustrates the class of dosage forms in which either IVRT or IVPT studies are recommended. Based on this data at least, we see that creams and ointment appears to have the highest percentage of increase, or high percentage of either IVRT or IVPT studies, which may be due to the complexity of these dosage forms. In this slide, I would like to illustrate some key attributes of IVRT and IVPT, and I will touch briefly on a few of them. In terms of receptor solution, it is recommended that physiological solution are typically used for IVPT studies, while aqueous or organic media are utilized for IVRT. Regarding the membrane, we typically recommend that IVRT should be conducted with synthetic membrane, while IVPT uses human skin. During these studies, we expect that occluded condition be used for IVRT to prevent metamorphosis or drying, while non-occlusion condition should be used for IVPT in order to mimic the physiological conditions of the skin. The last point I will mention here in this slide is regarding variabilities. We tend to see relatively consistent variabilities across IVRT studies, while for IVPT, some degrees of intra-interdonor variabilities are seen, but this can be minimized with adequate control of study procedures. For this slide, I would like to draw the attention of participants today to how these in vitro studies should be documented and organized to enhance the quality of ANDA submissions. We see in many ANDAs where the method development, validation, analytical, and pivotal studies are combined in one long report. The recommendation is to have separate individual reports for each study for ease of understanding of how each study was conducted and constructed. The individual reports should be organized in ECTD Module 5.3.1, and the summary tables, of course, included in Module 2.7. For this slide, I attempted to group together the common issues that we see in both studies. In some cases, we tend to see we don't see suitable details submitted for apparatus qualification and method control. We see inadequate data are submitted for validation of method parameters, as well as the discriminatory ability of the method are not evident from the submitted results. Additionally, we see details not included for data analysis results, as well as raw data are missing. So we encourage applicants to please consider these points as they put together their ANDA submission to the agency. So for now, for the next 4 slides, I will focus on some very specific observations, the expectations, and the points to consider for IVRT studies. However, please note that the observations and considerations mentioned here in this presentation are not exhaustive. The first validation category I will discuss is the apparatus qualification. We tend to see insufficient information or data to justify the selection of the release apparatus being submitted to the agency. Per USB 1724 chapter and FDA guidance, data to qualify the apparatus, such as the efficient area of orifice and volume of receptor solution that are generated in the in vitro BE site, should be provided. Also, if available, manufacturer's measurement should also be included. However, we expect that the calculation of your release rate should be based on measurements from the study site. I have highlighted 2 observations here for membrane qualification. First, we tend to see inadequate studies submitted to support the choice of membranes, such as membrane inertness. To qualify the membrane, studies may include the incubation of drug of study without membrane to monitor for drug loss not related to membrane binding, and incubation with membrane to assess any decrease. Also, physical description of membrane before and after the study duration is also valuable to the assessor. The second point I would like to make here is that details regarding membrane treatment and whether the membrane attained equilibrium before dosing occurred are not documented. Applicants are encouraged to follow the recommendations from the U.S. Pharmacopoeia and provide scientific rationale if the techniques are different. For receptor solution, at a minimum, data to show the solubility, stability, and linearity of release rate among replicates in receptor solution should be included in the submission. These studies should reflect the maintenance of steady-state release kinetics among replicates. For dosing procedures, if the selected deficient apparatus is based on USP 1724, we recommend that the dosing procedures outlined within should be utilized. We sometimes observed the use of unconventional dosing procedures. By that, I mean when dosing procedures are different from that in the chapter for that particular selected deficient cell. This is highly discouraged as it may introduce additional variability in the IVRT studies. Also, we encourage applicants to provide details on how dosing was done and synchronize with sampling time for each successive deficient cell. This will ensure that the precision of the sampling intervals are accurate. The next the next 2 I'm discussing here today is regarding sampling duration, sampling times, and robustness. We tend to see inadequate supportive data or evidence reported for the selection of study duration or sampling times. For example, if the sample duration differs from what is recommended in the USP chapter 1724, scientific rationale should be provided. However, it is worth noting that an optimized IVRT method should have study duration that can appropriately capture the steady-state release rate for both the test and reference products, for example, between 4 to 6 hours. IVRT, is a somewhat compendium method, at least for topical dermatological products. So it is expected that data to support the robustness of the method with predefined acceptance criteria should be provided. To determine robustness of the method, studies such as variations in temperature, dose, volume, receptor solution, and missing rate can be done. The last and definitely not the least validation category I would like to discuss for the discriminatory ability of IVRT is the discriminatory ability of IVRT. It is expected that details regarding the composition and preparation procedures for the altered formulations that are used for these studies should be provided. The altered formulation used for sensitivity and selectivity, which should be plus or minus 50% variation in strength from the nominal strength of 100%, are recommended. We think that this strength level should be able to demonstrate a reasonably discriminatory method. For supplemental selectivity, we have seen cases where multiple attributes are changed for altered formulation. We encourage that you consider changing one attribute at a time. For example, either a change in one critical excipient amount or change in manufacturing process is preferred, and not both changes at the same time. Also, note that selectivity should be demonstrated both qualitatively and quantitatively. Qualitative results such as individual concentration, cumulative amounts, and all plots for all the formulations assessed should be submitted, while a quantitative demonstration of inequivalence using the method recommended in the USP 1724 chapter is also expected. The final phase of the presentation will provide again some examples of observations, considerations, and expectations for IVPT studies. Again, please note that the list of items mentioned here in this presentation are not exhaustive. Just like in IVRT, insufficient data are provided to qualify the diffusion apparatus. In general, we typically see vertical diffusion cells and flow-through diffusion cells used for IVPT studies. However, regardless of the apparatus that is used, it is valuable to the assessor to provide information or data regarding the selection, as I've previously discussed during the IVRT studies, during my IVRT discussion. In some instances, data to support the selection of target dose, amount, and dosing procedures are not provided. It is expected that data to show accuracy and precision of your dose amount applied are submitted. By that, by what method dose was spread and how the dosing times were staggered and synchronized with sampling times should be appropriately documented and submitted to the agency. Dose duration and sampling time selected should be able to identify the maximum flux and the decline in flux. The precision and accuracy of sampling at different time points should be determined, and the data should be submitted to FDA. For the next slide, I'm going to focus on scheme validation. Regarding that particular category of parameters, we tend to see inadequate information for, say, skin processing and preparations. We expect that details such as skin handling, the thickness of distribution, equilibration, temperature measurements should be provided to the agency. Also, we encourage that applicants consider using the same anatomical area of skin for all studies. Another observation is the concern related to overhydration of skin. This is one fundamental difference between the IVPT and IVRT studies. For IVRT, it is typical to soak your synthetic membrane in receptor solution before mounting on the apparatus. This is not the case for IVPT, since overhydration of skin can compromise the permeability of the skin. So consider thawing your skin and briefly rinsing in warm water prior to use. Again, we sometimes receive ANDAs with insufficient description on how the skin barrier integrity was measured. Please, we encourage that you provide very detailed information on how the instrument was calibrated, outline pre-specified acceptance criteria, if possible with supportive data to be able to discriminate between skin sections that are compromised and competent. One key observation for receptor solution I would like to point out here today is the use of chemical agents, for example, organic solvents in the receptor solution. These have the potential to alter the permeability or may even destroy the barrier properties of the skin. If there are concerns of solubility, for example, in hydrophobic drugs, you can consider maybe adding 0.1% or 0.2% of olei ether, which has been demonstrated to not have any impact on skin permeability, to kind of increase the solubility of your test article. For IVPT sensitivity studies, applicants are encouraged to use sufficient number of donors and replicates to conduct the study and to submit all results, including the individual data and plots for all replicates for each and every single donor. IVPT study requires also a pilot study design as part of your overall assessment. So in this parallel design, we expect that the studies are conducted using your proposed test formulation, the reference product, and altered formulations. This pilot study is designed in such a way that it may help you determine the number of donors appropriate for the pivotal study, as well as demonstrate the selectivity of the method with the permeation profiles expected to be different between the test and reference product and the altered formulation. Some of the observations we see in the submissions are that the flux and cumulative permeation results and profiles for each donor are not reported or properly formatted for each deficient cell at each time point. We expect that details describing summary statistics for intra-donor and inter-donor average should be submitted. Again, we expect that overall, not just in pivotal study, that good documentation practice should be adopted throughout your study, which will allow for the submission of clear and detailed study reports to the agency, and also allow the assessor to hopefully reconstruct and understand your train of thought when we are conducting these studies. The next two slides, I'm going to focus on some observations and what the expectations are for IVPT pivotal study. It is normally recommended that whenever possible, the same batches of drug products should be used for all studies to demonstrate BE. That the details of study design, including, for example, documentation related to the control of your method parameters, should be provided. And we expect that the calculation of your flux should be based upon the precise empirical measured volume of that specific deficient cell, which may vary between individual cells. Finally, for this slide, please note that exclusion of data that are generated during pivotal study is generally not advisable without proper documented protocol violations or experimental errors. For IVPT study, it is not uncommon to see inter and intra-donor variabilities in this kind of studies. However, this can be minimized by adequate control of study parameters. In cases where abnormally high variabilities are observed, it is recommended that applicants check the method parameters and try to understand the factors that may be controlling permeation for their test article. In other situations, we also observe that the variabilities are inconsistent across studies. For example, validation data may show low variability, while the pivotal study shows high variability. When this occurs, it is helpful to provide the FDA with detailed explanations and possible rationale for this occurrence. In terms of data presentation for the pivotal study, we expect the qualitative and quantitative comparison of flux and cumulative permeation data to be submitted. This should include, for example, individual profiles and average profiles with and without error bars. The final point I would like to discuss in my presentation today is regarding the analytical validation for the sample analysis. We expect that a properly validated analytical method based on FDA guidance or ICH guidelines should be used for sample analysis. It is worth noting that the calibration curves that are selected should please cover the concentration range as well as relevant QC samples should be utilized. One last point is that the stability of drug of study in the receptor solution should be determined by using the highest relevant temperature. In conclusion, proper understanding of factors controlling release or permeation can guide the improvement of IVRT and IVPT submissions. Applicants should endeavor to optimize and validate methods to be discriminatory and reproducible. And a well-controlled IVRT and IVPT method should be used to conduct the pivotal study. And special attention to FDA guidance documents and considerations of the observations discussed here and in FDA workshop conducted for complex products will help improve quality of future submissions. Finally, I believe that FDA and applicants working together will help extend resources, reduce number of review cycles, and achieve faster approval. Now on to the challenge question. Question 1. Change of multiple critical attributes for an altered formulation is appropriate when conducting supplemental selectivity study for IVRT. A, true. B, false. I'm sure most people got this right. The answer is false. Next challenge question. Which of the following statement is not true? IVPT statistical analysis is determined by comparing test and reference products using Wilcoxon rank, Mann-Whitney rank test. IVPT is usually conducted using physiological media with microbial agents. IVPT endpoint parameters are the rate and extent of active ingredient availability at or near site of action. D. IVPT statistical analysis is determined using 90% confidence interval of log transform Jmax and cumulative amount permeated using average BE or reference scale average BE. The answer is A. Special thanks to my colleagues listed here for their contributions to this presentation. To the Division of Bioequivalence 2, the Office of Bioequivalence, and the Office of Research and Standards for their continued efforts towards developing consistent guidance recommendations for this in vitro test that could increase the efficiency of topical product submissions in ANDAs. Thank you for listening. I will be back in the Q&A section as a panelist to answer any question that you may have. Thank you.
Good afternoon, everyone. My name is Tannaz Ramezanli, and I'm a pharmacologist within Division of Therapeutic Performance 1, Office of Research and Standard. Today, I'll be talking about theoretical principles and some scientific and practical considerations for conducting in vitro release test or IVRT. This presentation reflects my views and not the views of the agency. The learning objectives for this presentation are first to describe considerations for IVRT study design when used as a component of characterization-based bioequivalence approaches, and describe considerations on the current thinking related to IVRT method development and IVRT method validation. While the focus of this presentation is the IVRT studies that are done as part of the characterization-based BE approach, the fundamentals of IVRT and most of the theoretical or practical considerations, especially related to method development that are discussed here, can be applied to all IVRTs for topical products and not just the ones that are conducted for BE purposes. IVRT is a compendial performance test to study the arrangement of matter. IVRT for semi-solid topical products are discussed in USB chapter 1724, and there are multiple apparatus that are introduced in this chapter that can be used for the IVRT study. However, what I will discuss in this presentation is the IVRT in the context of vertical diffusion cell, which is the most commonly used apparatus for the IVRT study. The basic concept of IVRT is that the dosage form is applied on the membrane that was mounted on diffusion cell, and the drug is released from the dosage form through the membrane, gets into the receptor solution, and by sampling the receptor solution, we can measure the drug that is released and calculate the drug release rate. There are three main phases for the IVRT study, especially when it's conducted as part of a characterization-based approach. The first phase is IVRT method development, next is IVRT method validation, and last is the IVRT pivotal study to compare the release rate of the test and reference product. As I mentioned earlier, the method development is the first phase and is exploratory in nature, and is where you need to select method parameters and optimize them. You can utilize a preliminary developed HPLC analytical method for sample analysis, and the release rates that are conducted during the IVRT method development are not expected to be analyzed using a validated analytical method. There are multiple method parameters that are evaluated during the IVRT method development, and the most important ones are selecting receptor solution and the membrane. And the sequence of these selections are: first you need to select the apparatus that you're going to use for the IVRT study, and it needs to be compatible with the conceptual designs that are described in USP chapter 1724. And next is selecting the receptor solution and evaluating it for your drug product and your IVRT method. And the next parameter after selecting the receptor solution would be the selection of the IVRT membrane. The receptor solutions that are used in the IVRT studies are typically hydroalcoholic mixtures that are selected based upon the solubility and stability of the drug in them. Because many of the topical products contain hydrophobic drugs, the receptor solutions contain alcohol or organic solvents. In order to qualify the receptor solution, you need to evaluate the solubility of the drug in them and make sure the sink condition is maintained throughout the IVRT study. You also need to evaluate the stability of the drug in the receptor solution at the temperature to which the drug is exposed to during the IVRT study. And in addition, you need to demonstrate acceptable linearity and precision of the resulting drug release profile using the selected receptor solution. So what is the acceptable linearity for an IVRT profile? Well, although historically a minimum linearity or R-square value of greater than 0.9 across the IVRT duration has been recommended, linearity of the drug release rate across all time points should ideally have an R-square value of greater than or equal to 0.97. And this is the value that you should target during the IVRT method development. Next, I'm showing some examples of the IVRT profiles where even though the R-square values are greater than 0.95, we see some deviations from the steady-state release kinetics and some parts of the IVRT or some curvature in the release profile. The first example, there is a curvature in the earlier part of the profile, and the first time point specifically doesn't seem to be taken when we have a steady state release, and the release rate here at the 30 minutes seems to be different than the next few time points. But R-square value is approximately 0.97. In the second example, we again see some curvature in the middle, and the last time point seems to have a lower release rate, but the R-square value is 0.96. And the last example clearly shows that the last time point has a lower release rate and is not at the steady state release anymore. And one reason behind such observation is that the solubility of the drug in the receptor solution limits the release kinetics and causing a reduction in the release rate at the last time point. So in this situation, it may be appropriate to evaluate a different receptor solution. So again, even though we observed such thing at the last time point, the R-square value is 0.96. So what we can conclude from these examples is that the R-square value may not be a sensitive criterion to evaluate the steady-state release kinetics, and sometimes it's much easier and more helpful to plot the data and plot the release profile to make sure that all the selected time points are actually selected when you're at the steady-state release. For well-developed and suitably controlled IVRT methods, you can routinely observe the R-square value of greater than or equal to 0.99. Dose depletion is expressed as the percentage of amount of drug released from the applied dose. Per Higuchi equations, the steady-state release kinetics is assumed when dose depletion is less than 30%. But does that mean that if dose depletion is more than 30%, the IVRT is invalid? Well, not necessarily. A dose depletion of more than 30% may be acceptable if the release rate remains suitably linear. So if you run an IVRT and let's say at 3 hours you observe dose depletion above 30%, for example 40%, you should not truncate the IVRT if you have steady-state kinetics beyond 3 hours, even though the dose depletion is more than 30%. But if you are in a situation where you observe that a decrease in the drug release rate at the last time point, like here, and deviation from the steady-state kinetics, and your dose depletion is also high, then you would need to optimize the method parameters or even considering applying higher dose amounts to obtain appropriate release kinetics during the IVRT. And if, for example, you have sufficient time points and duration before this last time point, you may be able to exclude that from your calculation, and here by excluding the last time points, we have improved the R-square value to 0.99 and have significantly increased the release rate. The IVRT membranes are typically filtered membranes that are selected based upon their effective pore size and inertness to binding to the drug. The pore size is often 0.5 micrometer plus minus 0.3. This is a suitable pore size, and it is selected based on the viscosity of the dosage form. The membrane binding study is one of the tests to qualify the selected IVRT membrane and is described in the acyclovir cream guidance. The membrane should also have chemical compatibility with the receptor solution, especially when using strong organic solvents in the receptor solution mixture. It is important to make sure the chemical compatibility exists between the selected membrane and the receptor solution. Another consideration is membrane equilibration prior to dosing in every IVRT study. That means that the membrane is mounted on a diffusion cell and it is in contact with the receptor solution, and the membrane temperature of the diffusion cell has been equilibrated and stabilized at 32 degrees Celsius plus minus 1. The apparatus usually can control the diffusion cell thermoregulation the way that you can set it the way that the surface of the membrane is maintained at 32 degrees Celsius, and this is a temperature that should be monitored and controlled during the IVRT study. This temperature is meant for topical dermatological drug products, but when you're conducting IVRT for the let's say a vaginal or rectal semi-solid products, then you need to target the membrane temperature of 37 degrees plus minus 1. A typical IVRT study should have a duration of 4 to 6 hours, and it's unconventional for the IVRT sampling time points to be selected within the study duration of less than 4 hours. There might be cases where, for example, you're working with a slow-releasing ointment and it may take longer to get to steady-state release kinetics, so you may end up running a longer IVRT study. But for most of the cases for semi-solid topical products, 4 to 6 hours IVRT is sufficient. And if you're running a shorter IVRT study, like a less than 4 hours, you should be able to justify why you could not run a longer IVRT study. And as I mentioned earlier, the dose depletion values of more than 30% are not a valid and adequate justification to truncate the IVRT study. Sampling frequency and number of sampling time points are also important. So for a typical IVRT study, 5 to 6 sampling time points should be sufficient, and the frequency should be the way that these sampling time points are well spread throughout the IVRT duration and are selected at a time where you are at steady state release kinetics. So for example, in this IVRT study, the first sample was taken at 30 minutes, and you can see that the release rate from this interval, 30 minutes to 1 hour, is obviously different than the release rate throughout the rest of the IVRT study. And by removing this, the first time point, our R-square value has improved and the release rate, the slope value, has increased. So it is likely that the first time point was not at the steady state. So you need to make sure that for calculation of the release rate, you're using the time points where you're at steady state release kinetics. The sampling procedure should be qualified as well. The accuracy and precision of the receptor solution sample collection at each time point should be appropriately qualified, and your sampling procedure should be able to reliably collect a consistent volume of the sample from well-mixed portion of the receptor solution. And this qualification is something that the investigator should do it, and not it's not something that you rely on the manufacturer, the apparatus manufacturer qualification. The dose amount for the IVRT study should be an infinite dose or semi-infinite dose, and should be sufficient enough to completely cover the whole exposed area of the membrane, and should be sufficient enough to provide a steady-state release kinetics throughout the IVRT study. So while there is no cutoff value or specific range to define this infinite dose amount, your IVRT profile would speak to whether the infinite dose were used or not. And for example, if you use a dose that is on the lower side, let's say 150 milligram instead of a 400 or 500 milligram of the product, and in the middle of the IVRT study, let's say at 4 hours, you start to see decrease in the release rate and deviation from steady-state release kinetics, is not appropriate for you to cut down the study duration and submit a 3-hour IVRT study, while you could have optimized your method, applied higher dose amount, and have the infinite dose for the IVRT study. The dosing procedure for the IVRT should be selected based on the apparatus that you're using. USP Chapter 1724 describes dosing procedures that are appropriate for each apparatus that is introduced in that chapter. and it's important that you follow the recommendations in the USB chapter to use the dosing procedure. For example, for vertical diffusion cells that are the subject of these presentations, for many of them, especially the ones that are compatible with Model B and Model C, the dose should be applied on a membrane that while it is mounted inside the diffusion cell and was equilibrated with the receptor solution. Also, the dose application method should not impact on product microstructure. For example, if you're using a syringe to apply a dose, you need to make sure that the shear forces that the product is experiencing while dispensing from the syringe is not affecting the microstructure of the test or reference products. Another consideration for the IVRT study is the occlusion. The donor chamber should be occluded during the IVRT study per the recommendations in the USB chapter 1724 to prevent loss of any volatile components of the formulation. And that's because per Higuchi kinetics, a steady-state drug release may not be achieved when the dose is not occluded during the IVRT and dosage form undergoes drying and metamorphosis. As an example, here is the release profile for a topical gel product when it was done under unoccluded condition. And you can see after 1 or 2 hours, the release profile starts to deviate from the steady-state kinetics, and we no longer have the linearity. So that's because the formulation gets dried and undergoes metamorphosis. But when the same product evaluated using the same IVRT method under occluded condition, we were able to achieve the steady-state kinetics during the 4-hour study and obtain a suitable linearity of the release profile. An important component of IVRT is to show the discriminatory capability of the IVRT method that can be demonstrated using 3 concepts of sensitivity, selectivity, and specificity. IVRT sensitivity is the ability to detect changes in the release rate as a function of drug concentration in the formulation, and can be established by comparing the release rate of the test or reference formulation with that from a higher strength formulation, such as 150% strength, and a lower strength formulation that has 50% of the nominal strength. And in order to establish sensitivity, you need to show that with the higher strength formulation, you can obtain a higher release rate compared to the nominal strength, and with the lower strength, you can obtain lower release rate compared to the nominal strength. Sometimes there might be challenges associated with formulating a higher strength of a particular drug product. So allowance may be made if higher strength of test product is not feasible to formulate without substantial reformulation. IVRT selectivity is the ability of the IVRT method to identify that the drug release rate from higher or lower strength formulations are inequivalent with respect to the nominal reference strength. And this inequivalence needs to be established using statistical analysis that are described in USB chapter 1724. And either test or reference product can be used for the IVRT validation. IVRT supplemental selectivity is the ability of the IVRT method to identify that the drug release rate from an altered formulation that has the same strength as the nominal strength of the reference or test product is inequivalent to the release rate of the nominal reference strength formulation. In order to establish supplemental selectivity, you need to introduce changes in the formulation by either change in the composition, Q1 or Q2 change, or change in the manufacturing process in order to prepare a formulation that has a significant drug release rate compared to the original formulation of the test or reference product. And the supplemental selectivity also needs to be established using statistical analysis. Precision and reproducibility are another component of IVRT validation. You need to conduct a minimum of 3 independent IVRT runs with 6 diffusion cells for each in order to show inter-run and inter-run precision and reproducibility, and calculate the release rate or the slope for each diffusion cell for these runs, calculate the mean standard deviation and percentage CV among the slopes across all runs, and a minimum percentage CV of equal to or less than 15% is recommended to establish precision and reproducibility. IVRT specificity is the ability to accurately monitor the proportionality of changes in the release rate as a function of drug concentration in the formulation, and can be illustrated by plotting the linear relationship of the release rate versus the nominal strength. To be considered suitably specific, an IVRT method should be proportionality linear in response to the difference in the release rate, and the acceptable linearity and R-square value is more than or equal to 0.95. While there is no requirement for the slope value of this linear regression line, the slope should be high enough that the method can discriminate higher release rates and the lower release rates from the release rate of the nominal strength, which in this example, this IVRT method shows that it's able to do that. But in the second example, here the slope of the specificity curve is very shallow, and this method is not as discriminative as the first method. And even if we have a little bit higher standard deviation, we might not be able to establish selectivity and passing the statistical test to show the inequivalence of release rate of the 150% strength with that of 100% strength. And even though it shows sensitivity and the perfect linearity for the specificity curve, but this method is not as discriminative as the first method. So that is another thing to take into consideration. So as part of IVRT validation, you should be able to show that the IVRT method is robust to small variations relative to the method parameters and apparatus. Robustness testing may encompass variation in the temperature, for example, plus minus 1 degree difference compared to the target temperature, which is 32 degrees Celsius. Variations in the dose volume or dose amount, for example, plus minus 10% difference in the dose volume. Variations in the receptor solution composition and pH, and variation in the mixing rate if the apparatus allows for that. So the average slope or the release rate of IVRT under these altered parameters should be within plus minus 15% of the average slope obtained from the precision and reproducibility runs. And if that's not achieved for one of the parameters, then that means it's very critical to control that parameter throughout the IVRT runs. And finally, the third stage or the final stage is the IVRT pivotal study that should be done in a manner that is compatible with USP Chapter 1724, where a single batch of the test product is compared with a single batch of the reference product. They are compared in parallel with 6 replicate diffusion cells for each. And the objective is to know that if the release rate from the test product is equivalent to that of the reference product or not. It is recommended that you use blinding procedure to blind the investigator who conducts the IVRT study, and once blinded, the test and reference product can receive random designation of A and B, and the dose and the product should be dosed in an alternative sequence. For example, if you assign them as A and B, the sequence can be A, B, A, B, A, B, or B, A, B, A, B, A, and you can refer to a cyclovir cream guidance for more information regarding blinding and sequencing and randomization. The cumulative amount of drug released at each sampling time point should be reported for each diffusion cells, and the release rate for test and reference products are compared using Wilcoxon rank sum Mann-Whitney rank test as described in the USP chapter 1724. In summary, you need to explain and justify the basis for the selection and optimization of the IVRT method parameters in the IVRT method development report. And there are method parameters such as membrane or a receptor solution that you will need to submit empirical data to justify how you optimize them. And there are other method parameters that may not be optimized using empirical data. So for them, you should explain how the conditions were selected for those method parameters in your method development report. And draft guidance on the cyclovir cream and USP chapter 1724 may provide a useful guide for the design, conduct, and validation of the IVRT studies. With that, I would like to acknowledge and thank my colleagues, teammates, and supervisors within US FDA. Here is the challenge question for my presentation. Which of the following should be established using statistical analysis? A, IVRT sensitivity, B, IVRT selectivity, C, IVRT specificity, D, IVRT robustness, E, all of the above. And the answer is B, IVRT selectivity needs to be established using statistical analysis. Thank you very much for listening to my presentation. And if you have any questions, you can submit it through the chat box, and we will respond to that during the Q&A session. Thank you.
Good afternoon, everyone. Thank you so much for the kind introduction. I am Priyanka Ghosh. I am an acting team lead within the Office of Research and Standards, Office of Generic Drugs. And today I'm going to talk to you about in vitro permeation testing, or an IVPT study. The specific learning objectives of this presentation are to discuss how and when an IVPT study can be utilized as a component of characterization-based bioequivalence approaches, and also discuss current challenges and thinking related to the design and conduct of such studies. I would also like to take this opportunity to highlight a presentation by Dr. Sam Raney at the IVRT IVPT workshop earlier this year in August, if you are interested in learning more about the fundamentals of an in vitro permeation test. So how can an IVPT study enhance our understanding within the scope of characterization-based approaches? Well, we heard about formulation sameness, we talked about physicochemical and structural characterization, and in vitro release tests. However, as the complexity of the dosage form increases, so do the risks associated with potential failure modes for bioequivalence. And an IVPT study allows us to evaluate the rate and extent of bioavailability of an active ingredient from the drug product as the dosage form undergoes metamorphosis. And thereby, it enables us to carefully understand how biphasic or multiphasic formulations would be expected to interact with the skin, and if there are any differences in these interactions between a prospective generic and a reference product. Now, how do we do these IVPT studies? We have the skin. We have the topmost layer of the skin, which is the stratum corneum barrier, which is the barrier to most active ingredients that permeate across the skin. And in an IVPT study, we take the skin, we use it as a membrane as opposed to the non-rate limiting membrane in an IVRT study. We apply a finite unoccluded dose which can undergo metamorphosis, and we have a physiologically relevant receptor solution. So once the API permeates across the skin, we collect the receptor solution, we quantify the receptor solution to assess the rate and extent of bioavailability of the active ingredient from the drug product. Now, in addition to this vertical diffusion cell system, a flow-through diffusion cell system is also often used for designing validated IVPT studies. In this case, you have a continuous flow of the receptor solution below the skin compared to the static receptor solution in case of the vertical diffusion cells. So how do you design the IVPT studies that are utilized for evaluation of BE or for comparing drug products? In the most conventional design, we compare the test product in multiple replicate skin sections from the same donor. We gather the data from using the study design that I showed you on the previous slide, and then we replicate the same study across multiple replicate skin sections for the reference product. Then we replicate this design across multiple donors in an effort to understand the population variability. And once we quantify the data, we look at flux versus time curves. As opposed to traditional pharmacokinetics where we are looking at concentration versus time, in case of these cutaneous pharmacokinetic profiles, we'll look at flux versus time. And these profiles allow us to evaluate the rate and extent of bioavailability of the active ingredient as the dosage form is undergoing metamorphosis. Now that we understand what kind of data we are looking for in an IVPT study, what are some of the key considerations that we need to keep in mind while developing these IVPT methods? Over the next few slides, I'm going to systemically walk you through some of the key considerations related to apparatus selection, selection of the skin source, selection of the receptor solution, assessment of barrier integrity, selection of the dose amount, dosing technique, and dose duration, selection of the study duration, the sampling schedule, as well as the methodology that can be utilized to develop an IVPT method that can then be used for evaluation of BE. So beginning with the apparatus selection, here we have some data from GADUFA-funded research projects where the studies were conducted in 3 separate labs across the world. We use the same product at the same dose, but we use different dosing techniques, different skin types, different thickness of the skin, different apparatus, different barrier integrity measurements. And what the data told us in each and every case is if we focus on the data, as long as we compared the test and the reference product, in this case, the Zovirax UX and the acyclovir 1A, using a very consistent methodology and a well-developed IVPT method, we were able to get very reliable and consistent results about the rate and extent of bioavailability of these drug products within each of these studies. There are, of course, differences in the absolute values of these absolute flux values as well as the shape of these profiles, and some of these may be attributed to the differences in the skin type or the dosing technique, and I'm going to talk about this more in the subsequent slides as well. Moving on then to the selection of the skin source, one of the key consideration here is, of course, we see in this example that we use different skin sources. So what is the key consideration here? The key consideration here is, again, to consistently use the same skin source from the same vendor or across all of your method validation and pivotal IVPT studies. And if you are either harvesting the skin yourself or if you are receiving the skin from an external vendor, it is very, very important to understand the skin harvesting procedures that were utilized. For example, how was the skin stored before it was shipped to you? How tightly the dermatoming was controlled so that you can assess the thickness of the skin and the range you would need to design a well-controlled IVPT study. It is also very important to develop skin preparation protocols before you load these skin sections onto your IVPT apparatus. And what I mean by that is, say, for example, you need to carefully consider how the skin is being handled once it's retrieved from the freezer. How long is it sitting on the bench top for thawing? how is the skin being cleaved, if at all. And very importantly, prevent any damage to the stratum corneum barrier as you are preparing the skin to load them onto your IVPT apparatus. And the last key consideration is, of course, you have to assess barrier integrity before you utilize any of these skin sections for an IVPT study in a given cell. However, before we talk about barrier integrity, let's talk about selection of the receptor solution. Of course, we need a receptor solution which would provide us with adequate solubility and stability. And what I mean by adequate solubility is ideally a solubility should be approximately an order of magnitude higher compared to the highest expected sample concentration during your IVPT study. However, we do want to note that you should still have a physiologically relevant receptor solution. In an effort to enhance the solubility, you should not add solubilizers such as organic solvents or specifically ethanol that may impact the barrier properties of the skin during the conduct of the IVPT study. On the other hand, you may be able to use something like OLEDS 20 at a concentration of 0.1% or 0.2% to be able to achieve adequate solubility for your analyte in your receptor solution. And last but not the least, of course, we recommend that you equilibrate your skin with your receptor solution on your apparatus of choice prior to assessing the integrity of that barrier, of that stratum corneum barrier. Now, how do you assess integrity? Well, historically, the three most common methodologies are transepidermal water loss, tritiated water, or transepidermal electrical resistance. And some of the key considerations as you are measuring barrier integrity or selecting an apparatus for measuring barrier integrity is to remember that depending on the methodology or the apparatus that you select, external factors such as temperature and humidity may play a role in your barrier integrity assessments. Also, it is important to define a cutoff criteria depending on experience in your lab and identify a cutoff criteria that can distinguish skin sections with a compromised barrier compared to skin sections which are expected to have an intact barrier. Now, once you have set up your diffusion cell, the skin, with the receptor solution and assessed the barrier function, the next step is, of course, to identify a dose. So how do you go about identifying a dose? This is just some example data sets from our Garufa regulatory science research where we show that when you vary the dose, and this is drying rate data on the top panel here, not IVPT data. This data shows us is thicker the dose for this gel and cream, longer it takes for these products to dry. And in this very small IVPT data set with 1 donor and 6 replicates, what this translates into is when it takes longer for the product to dry, you get a completely different IVPT flux profile for this drug product compared to, say, for example, the 10 milligram per centimeter square dose, which dries much faster and has lower bioavailability. So the key take-home point here is you may be able to conduct very simple drying rate experiments to be able to identify a dose that you may then want to evaluate during your IVPT method development studies. Another key consideration during the dosing is the dose application technique because it definitely impacts your IVPT flux profiles. This is a very small data set, again, with 1 donor and 6 replicates, where after dispensing the drug product with a positive displacement pipette, the dose was applied either with a spatula or a finger cord or a vial technique. And in this data set, for both the gel and the cream, it illustrates that there were no differences in bioavailability using either of these techniques. But the key take-home point here, again, is to identify and utilize a technique that you can consistently utilize to apply a very small, finite dose of your drug product throughout the duration of your IVPT method validation as well as your IVPT pivotal studies. And of course, we will talk a little bit more about the dose duration in subsequent slides. The other key consideration as you are developing your IVPT method is selecting the duration of the IVPT study, and what goes hand in hand with that is the sampling schedule as well as the sampling method. So this is an example with, I believe, a 1 donor 3 replicate study where in trial 1, you can see that the sampling schedule prevented us from being able to assess the true Jmax for this drug product. So we had to optimize the sampling profile, have more frequent sample at the beginning to adequately characterize not only the Jmax, but ideally the entire cutaneous pharmacokinetic profile as the dosage form is undergoing metamorphosis. And we encourage you to do the same as you are developing your IVPT method to identify a sampling schedule that would allow you to characterize the cutaneous pharmacokinetic profile, as well as a study duration that would allow you to do the same. Now, once you have identified your apparatus, your skin, your receptor solution, your dose, your study duration, your sampling technique, the next thing to do is to draft your IVPT method development report, of course. And you heard from previous discussions, but one thing we encourage you to do is chronologically document how you identified the study conditions. And this is especially important when you are using conditions that may be different from those that are outlined in the guidances. So the assessor is able to adequately understand and assess the scientific justifications for using these alternate study conditions. Now, once you have a well-developed IVPT method, the next consideration is to, of course, to validate the method. And like I did with the IVPT method development, I'm going to walk you through some of the key considerations related to IVPT method validation. This is not a comprehensive list, but some of the things that we have observed challenges with. The first thing is apparatus qualification. It is very, very important that once you have identified an apparatus, you qualify it during method validation, which may include assessment of the orifice diameter, assessment of the volume of the receptor solution, and so on and so forth. Other key consideration, of course, you have to qualify your skin. And by that, what we mean is you have to look at, you have to measure the barrier integrity and ensure that all skin sections fall within your inclusion criteria for barrier integrity assessment. You have to measure and you have to measure as well as report all of the skin thickness as well as the temperatures that you observe at the surface of the skin. For receptor solution qualification, you have to ensure that your receptor solution provides adequate solubility and stability for your analyte of interest throughout the duration of the IVPT study. And you also have to qualify the receptor solution sampling, because one key consideration for an IVPT study is often if you are not sampling from a well-mixed center of your IVPT receptor solution, because such small amounts of the active ingredient typically permeates in these IVPT studies, there may be errors in your analytical assessment that may lead to flux profiles that are not interpretable. So it is very, very important to develop an IVPT method that a receptor solution sampling method that allows you to consistently retrieve and quantify a sample that is from a well-mixed receptor solution. And of course, the sensitivity and the selectivity of the IVPT study, which I'm going to talk about on the next slide. One important consideration is for the method validation studies, please remember to utilize an validated analytical method for all of the studies that are conducted within the scope of method validation. Now, let's talk about the sensitivity and selectivity studies. So what is the role of the sensitivity and the selectivity studies? Well, with the sensitivity studies, we want to ensure that the IVPT method that has been developed has the ability to detect changes in cutaneous pharmacokinetics as a function of drug delivery. And the rationale for conducting this study is your method may be able to assess and show that products are bioequivalence, but we want to make sure that your method has the discriminatory ability such that it is able to detect changes in cutaneous pharmacokinetics when such differences exist. Now, you can assess the sensitivity either by modulating the dose, such as the example that I showed you before, with different thickness of the dose, especially for products which undergo rapid metamorphosis, such as gels and creams. For something like ointments, you may have to modulate the dose duration in order to establish the discriminatory ability of your IVPT method. Now, the sensitivity study is often conducted at the end of the method development phase. The selectivity study, on the other hand, is often conducted as a component of the pilot study, where you compare the test product, the reference product, and an alternate product. And for these, for the selectivity study or the pilot study, the studies are typically conducted in 4 to 6 donors, anywhere between 4 to 6 replicates per donor. And the goal of this study is, one, to assess the variability that exists in the bioavailability of the active ingredient from your reference product across the different donors that are utilized in such a study. And this allows you to identify the number of donors that you may need for the pivotal study, and also allows you to compare the test and the reference product, as well as differentiate those flux profiles from an altered formulation. Now, once you have appropriately validated your entire IVPT study, the next step is, of course, the conduct of the pivotal study. And I'm not going to talk about the pivotal study design, because we have already talked about how the IVPT studies are typically set up. But I do want to highlight this presentation by Dr. Elena Rantu, again at the IVRT IVPT workshop in August of this year, because in addition to the statistical methodologies that have been reflected in our product-specific guidance in the past, where a balanced data set had to be utilized for statistical analysis of IVPT data for assessment of bioequivalence, the presentation discusses how an unbalanced data can be utilized for assessment of bioequivalence as well. And the key thing to remember here is, we appreciate that sometimes aberrant data, such as the one shown in the example here, is observed during an IVPT study. The important thing to remember here is, whenever you are excluding a data, it must be adequately supported by protocol violations or experimental errors that happened during the conduct of the study. And we acknowledge that there are some key considerations related to handling of aberrant data without documented protocol violations and experimental errors that require further research, and is an area of active interest for the agency at the moment. However, the key thing to remember for the pivotal studies is to control your entire IVPT setup to minimize loss of data during the conduct of the study, and also to ensure that the variability is as low as possible from the methodologies that you utilize to conduct the study. So with that, I'll move on to my challenge question for today. We talked a lot about IVPT study. So what, in your opinion, is the role of the IVPT study as a component of the characterization-based approach? Is it to deformulate the drug product, to characterize the physical properties of the drug product, to quantify the release of the active ingredient from the drug product, or is it to understand the rate and extent of bioavailability from the drug product? Well, it's D. So you are all correct. So with that, I would move on to my summary, where I hope I was able to discuss with you today how an IVPT study is typically utilized within the context of these characterization-based approaches. For the method development studies, the key consideration is to systemically identify study conditions that are relevant for a given drug product and clearly document them within your method development report. For the method validation studies, it is very, very important to validate the study conditions and also establish the discriminatory ability of your IVPT method. And last but not the least, for the pivotal study, of course, it is very, very important to implement controls to minimize the variability and loss of data during the conduct of the study. With that, I would like to acknowledge our entire team within the U.S. Food and Drug Administration, Dr. Sam Raney, Dr. Eleanor Rantu, Dr. Tannaz Ramezanli, Dr. Mangmang New, Dr. Megan Kelchen, Dr. Ing Zhang, Dr. Hiren Patel, Dr. Markham Luke, Dr. Lei Zhang, and Dr. Robert Lionberger for their contributions to all of the research and data that I had the privilege of presenting today. I would also like to acknowledge our research collaborator who contributed to a lot of the data that I utilized to demonstrate key considerations during the presentation today. And I would like to thank all of you for your attention, and I look forward to your questions during the Q&A this afternoon. Thank you.
Thank you to all of our presenters for all of the great presentations today. We'll now enter into our last Q&A panel of the sessions. Our first group of questions will be directed to Dr. Josephine Aimiuwu. Here's the first question for Dr. Aimiuwu. Can IVRT and or IVPT be potentially used to support the BE of a topical suspension product? On slide 5, topical suspension is not included in your summary of topical ANDAs with IVRT and or IVPT.
Thank you for the question. To potentially demonstrate BE for topical suspension products, yes, IVRT and only or IVPT can be used. However, to my knowledge, at least based from the ANDA submissions that are included in my data in slide 5, there are very few ANDAs that have submitted these types of studies. So we typically see clinical endpoint studies or vasoconstrictor studies in ANDAs. Thank you.
Thank you for responding to that question. We have another question for Dr. Aimiuwu, and here's the question. Does the IVPT method need to be validated before performing the pilot study?
Again, thank you for the question. For IVPT method, for the pilot study, you don't need to validate the IVPT method before you conduct such study. However, it is recommended that your IVPT method development has been conducted and some parameters have been determined before the pilot study is done. As I discussed in my presentation, pilot study is conducted in parallel using the proposed test formulation, the reference products, and altered formulations. So this pilot study, the essence is to help determine the number of donors that may be appropriate for the pivotal study, as well as demonstrate the selectivity of your method, which is a part of the validation of your IVPT method. Thank you.
Thank you for responding to that question. We have another question for Dr. Aimiuwu. And here's the question. When performing IVRT sensitivity studies, should the lab scale 50 and 150% batches be compared to a lab scale 100% batch, or should they be compared to the exhibit scale batch? For some products, the lab scale batches do not completely represent the exhibit scale product, and they have different release rates.
Thank you for the question. We tend to see these questions being asked in most of the workshops that have been done by FDA. So in general, when performing IVRT sensitivity studies, the agency would prefer, if possible, that exhibit scale batches are used. However, we understand the challenges and limitations in resources. So the lab scale batches could be utilized. We know that the goal of sensitivity studies is able to show discriminatory ability of your method from similar formulations by comparing the release rate from the test formulation with that with two comparable formulations in which the concentrations of drug has been altered, in this case with a higher strength, which is the 150% of the nominal reference concentration, and the lower one, which is the 50%. In other case, the use of lab batch or exhibit batch, we recommend that the composition and procedures for preparation of these altered formulations be reported, and it should show that similar equipment and at least manufacturing conditions are used for both the lab batch and exhibit scale batch that we expect should be used for the pivotal study. Thank you.
Thank you for responding to that question. We had a few more questions coming in for Dr. Aimiuwu, and here's the next question. There are general concerns from applicants regarding the number of bioequivalence deficiency comments pertaining to the IVPT, IVRT, especially IVPT studies received from the agency. Any advice on how to bridge these deficiencies?
Again, thank you for that question. The agency understands the concerns and challenges in addressing several deficiency comments that we communicate after the evaluation of your ANDA. And so I believe a way to minimize the number of deficiencies, we encourage applicants to submit detailed information of their study designs, as I've showed in my presentation results, provide explanations and rationales for any deviation from FDA guidance recommendations or USP chapter, especially in the case of IVRT, for all the steps that you utilize in conducting your in vitro studies. Because I believe this will assist the bioequivalence assessor to understand the data that you present to the agency, especially in this case, also for IVPT study, we tend to see like we don't have a lot of method. The methods are not well standardized yet. So when the applicants adopt good documentation practice throughout their in vitro study conduct, this will allow for the submission of very clear and detailed study reports to the agency. This, I believe, could help reduce the number of deficiency comments to the applicants. Thank you.
Thank you for responding to that question. We had just a couple more questions for Dr. Aimiuwu. And here's the next question. Is the determination of inequivalence between release rates using the statistical approach described in USP General Chapter 1724 sufficient to demonstrate supplemental selectivity for IVRT, or do additional qualitative results such as individual concentrations, cumulative release, and plots recommended to be submitted to the agency?
Great question. Thank you very much. In addition to the determination of inequivalence using the STAT approach that is described in USP chapter, we encourage that applicants submit very detailed plots, their qualitative results such as the individual concentrations, the cumulative release and plots, and that will help support the selectivity of their method. The release profiles, we further explain the kind of release trend, whether it's higher or lower consistently between the nominal formulation or reference formulation and the altered formulation. Thank you.
Thank you for responding to that question. We're going to change panelists, and right now we're going to go down to Dr. Tannaz Ramezanli, and here's the question for Dr. Ramezanli. Should one assess the linearity of the IVRT profile using individual cell data or average data?
Thank you for the question. It's always helpful to plot the individual cell data and use the individual cell data for your analysis, and especially like during the validation and the pivotal study, if you plot them individually and you may see that if there is something wrong with one of the cells that have changed the overall profile, and if you just look at looking at the average profile, you may not see that which cell resulted to that deviation. So it's always helpful to use to plot the individual cell data and look at the linearity and other aspect of the IVRT. Thank you.
Thank you for responding to that question. And we have another question for Dr. Ramezanli. And here's the question. In terms of manufacturing the altered formulation to demonstrate supplemental selectivity, the first speaker mentioned that multiple parameters such as composition and manufacturing process should not be considered, while the second speaker mentioned that it's okay to alter both parameters. Would you please provide clarification?
Sure. Thank you for the question. So I think the objective for supplemental selectivity, and as it says by its title, it's supplemental to your actual selectivity, is to show that you can compare your formulation with a different formulation of that API at the same drug concentration, and your that has a different release rate, and your method is able to discriminate the release rate of your formulation from that altered formulation. Sometimes by a simple Q2 change or a manufacturing change, you may be able to come up with a altered formulation that has a different release rate compared to the original formulation, and sometimes it is more challenging and you need to introduce multiple changes, or even we also recommend sometimes using a different dosage form. For example, for a cyclovir cream 5%, you can use a cyclovir ointment at 5% as an alternate formulation for supplemental selectivity. And if you're using a dosage form, obviously you have a different components in the ointment compared to cream and have different manufacturing process. So I want to emphasize that the main objective is to have a formulation at that same API concentration that have a discriminated release profile and release rate compared to your original formulation. Thank you.
Thank you for responding to that question. We're going to move on to our next panelist, Dr. Priyanka Ghosh. And here's the question for Dr. Ghosh. What about drugs which do not show Jmax and are not meant to have this due to target site on the skin? What additional data or information can be submitted?
Thank you for the question. I interpret the question to mean that in some situations we observe a cutaneous pharmacokinetic profile where we see the observe the Jmax at the last time point, because Jmax is the highest flux that we observe within the pharmacokinetic profile. So there is always a Jmax, but in some situations we may not have a decline in flux after the Jmax. So what should be done in those situations? Well, there are options. If this is a drug product that is not expected to undergo rapid metamorphosis, such as an ointment, you may need to consider alternate study designs where you may need to remove the drug product after a certain length of application in an effort to obtain a profile that provides you with not only the Jmax or the highest flux, but a decline in your flux profile from that point. Alternatively, you may need to do a little bit more with method development to identify what kind of study conditions, such as the dose application technique, the thickness of the dose, etc., can help you to obtain a pharmacokinetic profile where you can obtain a Jmax and then decline in flux similar to what you typically observe in pharmacokinetic profiles in for systemically acting drugs. Thank you.
Thank you for responding to that question. We're going to move on to another panelist, and this is for Dr. Sam Raney. And here's the question. What are the situations where an IVPT study is required for ANDA submissions? Is the IVPT study a requirement for any NDA or IND submission?
Okay. So I guess I should clarify, maybe it's semantics, but an IVPT study is not required for an ANDA submission. But it is submitted in many ANDA submissions to support a demonstration of bioequivalence. And it is also submitted in NDA submissions in certain instances. But there is a distinction where in an NDA, you are demonstrating safety and efficacy, whereas in an ANDA, you're demonstrating bioequivalence. And so the role that an IVPT study plays in an ANDA is to really support that demonstration that there's no significant difference in the rate and extent to which the drug becomes available at or near the site of action in the skin. And so the instances, the situations where an IVPT study becomes useful for that demonstration is for dosage forms where they are multiphasic vehicles, like emulsions, where the complexity of the dosage form and the number of different interactions, the types of different interactions between the dosage form and the skin are a little bit more complex, and there's more opportunities for something to potentially represent a failure mode for bioequivalence. And so those multiphasic vehicles, typically emulsions, are the ones where IVPT studies can be really useful for ANDAs.
Thank you for responding to that question. Well, we've just got a few more minutes left, and we're going to bring in our last panelist, Dr. Markham Luke. And here's the question for Dr. Luke. What are possible future avenues to integrate new pre-ANDA science with decision-making for generic drug applications for topical drugs?
Thank you, Jeff. Good afternoon. So the answer for that is that we continue to assess new technologies as they come in. And it's a knowledge management exercise of matching that technology or those new tools for products that are in our pipeline for us to develop product-specific guidances, approaches for bioequivalence evaluation, etc. And this is broadly applicable not just for topical products, but having those tying the tools and the research to the specific product areas that we're interested in. Of note, one of the most important things from my perspective is having good people like the folks who have presented at these last two panels, who can tie the pieces together and who have that understanding of the technology, have sufficient education and knowledge to pull together and bring about those particular changes to our policies. And so I wanted to commend our folks in the last two parts of this session for the fine presentations that they've done in the area of topical products. Thank you so much.
Well, that's all the time we have for questions in our Q&A panel. We certainly would like to thank all of our presenters and panelists over the last 2 days for all the great